Hi, my name is Aaron Sutter. I'm the CSO of the Twist Biopharma vertical here at Twist Bioscience. And today I'm going to tell you about how we utilized our antibody phage display libraries within the team to actually discover antibodies to both the SARS-CoV-2 S1 protein as well as ACE2. So I always say the best companies out there really understand the one thing that they're really good at. And for Twist, that's our ability to print DNA using our silicon platform shown on the right. So it's basically a fantastic technology that allows us to print large pools of individual oligos up to 300 base pairs in length, and we can actually print up to 1 million different oligos on this chip. And again, we can use this platform to make all kinds of custom DNA products. In particular, custom clonal genes is our top product that we use that you can go online and order on our website. You also have custom NGS enrichment kits for upstream of sample prep for NGS sequencing. But also we can use these large pools of oligos to create DNA libraries, which is actually central to the Twist Biopharma vertical here at Twist. So how do we use oligo pools to do that? So if you think about an antibody domain, either a heavy chain or a light chain domain, it's basically a collection of 3 different loops. We can synthesize large pools of oligos for each of these different loops and then just basically piece them together seamlessly to create a hypervariable domain. The reason why in my mind this is really game-changing compared to how people have made libraries in the past is that in the past people have used what's called a degenerate oligo to encode that diversity. Even though they oftentimes can get really close to what's seen in nature, they oftentimes can introduce unnatural mutations into their sequences or introduce errors into their library. Because we're using oligo pools, we can get a really high level of quality from our libraries, as well as, again, exactly match the CDR sequences that are seen in nature. We also can remove sequences up front that might lead to liability risks, such as isomerization, cleavage, deamidation, or glycosylation sites. And then finally, if we choose to, we can even insert motifs into those CDR loops as well. And so again, this is the technology that we're using now to create a whole suite of different libraries. I've coined the term a library of libraries to offer up to our customers and partners to help discover antibodies to different targets. And if you look at that library of libraries today, it's a huge breadth of different libraries, from a fully human Fab library called the hyperimmune library on the bottom right, and we even have a common light chain version of that same library. We have a whole suite of different VHH libraries shown on the top left. We also have a library that we call the structural antibody library, which is an scFv phage display library derived from all known antibodies that have ever been crystallized. And then again, we've also focused on difficult to drug targets, and we have a whole suite of different libraries directed against GPCRs, ion channels, and even carbohydrates. And so again, we take these libraries through traditional phage display workflow. Again, we pan our libraries against the either the cell-based or bead-based target. We do multiple rounds of panning, pick our clones, do ELISAs, sequence all of our clones, and then because we sit inside Twist, we can easily reformat all of our DNAs directly to full IgG or VHH-FCs, scale them up, and do high-throughput antibody production followed by binding and characterization assays. We're also pushing forward with a new alpha product within Twist, which is our ability to not only make huge numbers of DNAs that encode for antibodies, but also the physical antibody itself. Again, we've been doing this within the Twist Biopharma projects for quite some time now, and it's our vision that we want to push this out as another service that we can offer up to our customers. And certainly for this project, we use that ability to make antibodies either at the 1 mil or 8 mil scale to really quickly produce antibodies for testing so that we could do all the binding and functional data that I'll talk about today. So getting to the example I want to talk about today, which is finding antibodies to the SARS-CoV-2 virus, we really attack the virus by 2 different strategies. Of course, like everyone else, we're going after the virus itself by targeting the S1 protein, and so we have a panel of antibodies to S1 that I'll talk about today. But we also thought going after the receptor for the virus was also a potential therapeutic strategy, so we also found antibodies to the ACE2 receptor that also blocked the interaction with the virus. So as one of the libraries that we panned against the virus, we actually generated a COVID-19 specific library, and we did that in collaboration with Vanderbilt, where they actually gave us some sequences derived from a COVID-19 survivor. And we took that as an input data set to create a specific synthetic library that we panned and screened against the S1 protein. So as a whole, as an overview, how do we go after finding antibodies to the virus? So again, we used SARS-CoV-2 S1 RBD is one of our targets, as well as ACE2. We panned 3 different sets of libraries: our hyperimmune antibody phage display library, which again is a fully human Fab library, the COVID-19 library I just talked about, as well as several of our single domain libraries that I mentioned before. We did 3 or 4 rounds of panning against each target, picked a number of 3D 4-well plates and did phage ELISAs. We took all of those unique clones, quickly synthesized them inside Twist, and then used our high-throughput alpha product I talked about before to make large numbers and small amounts of antibody for initial testing. We put them through a battery of different affinity, receptor ligand competition, as well as developability assessment. And within 6 weeks, we actually had putative leads in hand. Of course, we put them through their traditional panning rounds with phage displays. So again, shown here are 4 different panning campaigns that we did. TB182, TB181, and TB182 are actually the S1 selections, and TB183 and 184 are the ACE2 selections. And what you can see is we're seeing nice enrichment of titers as we go to rounds 1 to round 4, indicating that the pannings are working and enriching for clones potentially against each of these 2 different targets. When we look at a whole and look at the sequence diversity of all the hits that come out of the ELISA screen we did, we see a large number of ELISA positive hits. And then on the inside of this wheel, we actually plotted how the sequences are related to one another. And as you can see, we see a huge breadth of different sequence families for the S1 binders that we found. On the outside of the wheel, we also plotted the ELISA signals from that, and we see really nice, strong signal-to-background ratios on the order of more than 10 for many of the S1 binders. And in addition, we also did a competition assay where we looked to see are the S1 phage clones actually competed off by ACE2, and for many of them shown in blue, you can see some competition with the natural receptor. So again, showing that potentially we have high affinity binding of our S1 antibodies that are actually competed off by ACE2. We did the same thing for our ACE2 binders. We see, again, a lot of different sequence families inside this phylogenetic wheel shown on this slide. On the outside of the wheel, we see nice signal-to-background ratios, again, on the order of 10. And then finally, we did the same kind of competition assay, but in this case with S1, and we see nice competition in a phage ELISA as well. We then took all of our unique clones, scaled them up using the high-throughput alpha product I talked about before, and then did a whole series of different kinetic assays using our Carterra LSA platform. So first and foremost, we looked at affinity of each of the antibodies to their respective targets, and then finally we did epitope binding, in particular for the S1 binders, to understand which portions of the receptor they bound to. So initially, we made a subset of our antibodies very quickly, and just to understand what kind of affinities we were seeing. And then for ACE2, we were shown on the left, we see a huge range of different affinities, monovalent affinities for our antibodies in the nanomolar, subnanomolar range. And then for S1, we also saw, again, a large number of clones that had nanomolar, subnanomolar binding. And this, again, was just a subset of the initial binders initially that we found. We then made and tested more, and as you'll see later, we have several clones that actually have picomolar affinity to both targets. If we directly couple the antibodies to the chip and then actually flow over the monovalent targets, we can get some really nice affinity data for the S1 antibodies for both S1 as well as S trimer that we purchased from Acro. And again, what we can see shown here on this slide is that we're seeing picomolar affinities for many of the antibodies to either S1 and or the S trimer, indicating we have very high affinity binding for many of our S1 antibodies to the target. Similarly, when we immobilize the ACE2 antibodies on the chip and flow over ACE2, we're also seeing, again, a high affinity monovalent binding in the picomolar range for many of the antibodies to ACE2. When we dive in a little bit further on some of the top leads that I'll talk about later, shown here are the top 3 leads that I'll spend some time on, TB182-3, TB182-4, as well as TB182-7. And as you can see, for either S1 and or S trimer, we're seeing picomolar to nanomolar binding for each of the three different antibodies. When we look more at specificity of these antibodies, in particular, the top 2 leads, TB182, 3, and 4, we wanted to see whether there was any cross-reactivity with the SARS-CoV-2 S1 protein, original SARS-CoV virus S1 protein. And we see that only those 2 antigens show any, those 2 antibodies show any binding to the original SARS-CoV virus. Shown here at the bottom are also some controls where we looked at the both acro antibody that we purchased as well as the CR3022 antibody, which again is a antibody that's originally isolated against the SARS-CoV virus. And as you can see, that antibody actually shows strong cross-reactivity between the original SARS-CoV virus and SARS-CoV-2, whereas our antibodies are actually specific for SARS-CoV-2. Another assay that we like to run with our antibodies is also an ELISA. This gives you, again, a broad range of different affinities of all the antibodies that we discovered. And as you can see here, and a lot of our top leads show really potent subnanomolar binding in ELISA. But of course, we have a huge number of other leads as well, shown here in this slide, that have varying levels of affinity based on this assay. And as you can see, our top inhibitors that potentially might be therapeutics are actually some of the best binders in this assay. And then we're also, again, pushing forward some leads that potentially are non-competitors, but high affinity, and those actually might be very useful for diagnostics. Similarly for ACE2, we can also see potent binding to, in this case, in a FACS-based assay. So again, Vero E6 cells are a monkey cell line that actually express ACE2, and we actually tested all of our ACE2 antibodies for binding to this cell line. And here you can see that a lot of our top clones, which are shown in green, show nice high affinity binding to the cell line using this assay. We also used a inhibitor assay derived also from acro, where we immobilize S1-RBD on the plate, and then we incubate our antibody with biotechnology ACE2 and see if we can inhibit the interaction with the S1 on the plate. And we see that a lot of our antibodies, both from S1 as well as ACE2, inhibit the interaction between S1-RBD and ACE2. We also developed a cell-based assay where we took, again, the Vero E6 cells, which express ACE2, and we tried to see whether we can compete off the interaction between S1-RBD and our antibodies for binding to the cell line. And what we see is that for our top S1 antibodies, TB182 and 3 and 4, we see nice, potent nanomolar IC50s in this assay, where we see that we can block the interaction between S1 and ACE2. And we see that the IC50s that we see from this actually are actually very comparable or even better than the CR3022 control as well as the acro control that we used. When we actually dive in a little bit further into the epitopes that these antibodies bind to, we can see that they actually bind to several different epitopes on the S1 protein. So the TB182 and 3 and 4 actually reside in this bin up here that actually has many of our potent inhibitors of the S1 ACE2 interaction. The acro antibody, which is also neutralizing, actually sits in its own bin, and the TB182-4 antibody actually bridges the acro antibody with that larger bin that includes TB182-3. What we also find is a lot of our high affinity antibodies that actually don't compete with that interaction actually sit down in this bin, which again shows us that potentially that we might be binding to another part of the receptor that we might be able to use for diagnostic applications that actually don't inhibit the natural interaction between S1 and ACE2. And then just to sum it up, we took all of that data, tabulated it here for S1, and as I'll have another table for ACE2, where we looked at, again, their KDs for both in an anti-FC capture, which I didn't show that data, as well as a direct capture on the LSA. We also looked at, again, ELISA bind that I showed before, inhibition in an ELISA-based assay, and then also inhibition in this cellular-based assay. And from all of this, we have two top candidates, TB182-3 and 4, for potentially for therapeutics that inhibit this interaction that could be potentially used to neutralize the virus. But we also have a whole suite of other antibodies that don't inhibit that interaction that potentially might be useful for diagnostic applications, and those are actually indicated by an underline on the far left. And then, as I said, for ACE2, we also have similar data showing that we have huge breadth of different sequences that go out that combine with high affinity to ACE2, and we see nice inhibition of the S1 ACE2 interaction derived from these antibodies as well. So just to summarize, we've now discovered a whole suite of different antibodies to both S1 as well as ACE2. They have picomolar to nanomolar binding to the receptor. We've shown that we can block this interaction between S1 and ACE2. We've done full epitope binding on the S1 side. And we plan to scale up these antibodies and actually test them further in neutralization assays, as well as assess to see whether we can actually make multivalent or bispecific versions of them to increase their overall avidity and potency. And then finally, we're following up by doing additional screens with some of our other libraries to see, in particular, for example, could we actually find some single domain binders as well to the S1 protein to add on to some of our initial constructs.